Hi folks, welcome back. Today I want to move on to discuss the next function of molecular biology, that being translation. And let me start by reminding you what that is using this slide. This is a slide I've used many times before, and it's the complete version of the dogma of molecular biology, the one the evolutionists don't want you to see. And just for a reminder, here's the incomplete version, the one that they teach everyone in just about every science class. And as I've told you before, this is intentionally and strategically left incomplete, because once you learn the complete version, you realize that the dogma of molecular biology is impossible to explain scientifically. So let's go back to that one here. Here's our complete version. And like I said, this is the one that's impossible to explain scientifically. So I won't go over all the rationale for that again. But what I want to use this for today is to point out that the process of transcription creates RNA from DNA, and we've already discussed that in a previous podcast. And the process of translation creates proteins from RNA. And the process of replication creates more DNA from existing DNA. And this process of translation is what I want to discuss today. Translation is the biological process where an mRNA, meaning messenger RNA, molecule is used to make a protein. Translation is an appropriate term because the language of DNA, copied through the messenger RNA, must be translated into the language of proteins. The process of translation is primarily accomplished through a structure called a ribosome. Ribosomes are made up of ribosomal RNA, rRNA, and ribonucleoproteins, proteins that are combined with RNA. The ribosomes are divided into two subunits, which together are called 80S. The smaller subunit is called the 40S subunit, while the larger subunit is called the 60S subunit. And that is actually made up of three other subunits. Ribosomes bind the messenger RNA with the help of things called initiation factors. And then they read along the messenger RNA molecule in a 5' to 3' direction, and they create a protein from the mRNA template. The mRNA is passed through the ribosome, similar to a ticker tape machine or maybe a conveyor belt going through an assembly line. And as it does, the ribosome decodes the information found in the bases. They translate the coded information from the bases into the corresponding amino acids. Like nucleotides to DNA and RNA, amino acids are the building blocks of proteins. They're the subunits that are linked together in varying orders to create specific proteins. However, there are 20 different amino acids that can be used to create proteins, where with DNA and RNA, they only have the 4 nucleotides. And if you look at this slide, you'll see the basic structure of an amino acid. They consist of a central carbon attached to 4 different groups in a linear pattern when we visualize it in 2D. The 4 different groups in an amino acid are a carboxyl group, which is this part over here. We have a hydrogen atom, which is that guy right there. We have a nitrogenous amine group, which is the NH2 right here. There's one exception with that NH2 group. There's one amino acid that only has an NH group there that's modified into a ring structure. And then we have this side group over here, the R. The first three groups are common to all amino acid. The side chain group, this guy over here, that's what makes each amino acid unique, since all of them are different for each amino acid. The secret behind the ribosome's ability to translate the information in the bases into amino acids is through the use of codons, where 3 bases are read at one time. Each 3-base codon codes for an amino acid, with a few exceptions. But the question here is, how does the ribosome know which codons call for which amino acids? And the answer is, it's because they are the Rosetta Stones of the genetic code. What they do is they pull out their Ovaltine decoder ring, and that tells them which codons go for which amino acids. You remember that? The Ovaltine decoder ring from A Christmas Story with Ralphie? Yeah, everybody remembers that one, right? Well, the truth is the ribosomes don't need Ralphie's Ovaltine decoder ring because they are the keepers of the actual genetic code, which shows which codons code for which amino acids. Each 3-base codon has an associated amino acid, with many having more than one. So, for example, and remember, RNA uses U, uracil, instead of T. So we're looking at RNA bases in three in a row, a codon. So like, for example, here we're showing these 3 bases and these 3, UUU and UUC. So those would both code for, that's phenylalanine. And then these, all of these 6 combinations, that's a leucine. If you look over here, all of these would be arginine, and so would these. This is a serine. This is a proline. These are more serines, etc., etc. So all of these combinations have an associated amino acid that it codes for. And there's a couple special categories, like these guys over here, these guys, and this one. They all code for stop codons. They tell the ribosomes where to stop the process of translation. And then we have one special category down here, AUG. That is both a methionine and a start codon in eukaryotes. It tells the ribosomes where to start translating. So when it comes to that, it will know to start, and the first amino acid that it will put in will be a methionine. Now, as I explain translation, I want to show you this 2D image to help you visualize the process. Here's the small and large subunits of the ribosome, which is, so we got those labeled there, right? This is the large one. And then the red in here, right? This is the mRNA being fed through the ribosome. The small subunit of a ribosome reads the codon ATG, like we just said, that's the start codon, and recruits the transfer RNA molecule, which is this, that's transfer RNA molecule, or tRNA. That contains the complementary 3 bases, these 3 right here, that would match the 3-base codon of the first one it starts with, the ATG. The ribosome recruits the tRNA with the correct 3-base key, the anticodon, that matches the 3-base lock, the codon, that it finds in the mRNA. This matching of these bases in the codons and anticodons follows the same complementary pairing of RNA and DNA, where A binds with U and G binds with C, and vice versa for both of those two. So on one end of the tRNA, you have the 3' anticodon RNA, while on the other end of the tRNA, you find the amino acid attached that matches their specific 3' anticodon code. And that's what I'm showing here, this circle up at the top that is attached to that tRNA. Therefore, there are different tRNAs for each of the 20 possible essential amino acids. And when the tRNA is complex to their specific amino acid, it is considered to be charged. They call it charged. When the appropriate tRNA is recruited by the ribosome, it's base paired with the 3' codon in the mRNA. This process lines up, so that's what I'm showing here. Here's a correct pairing, right? And here's a correct pairing, 3 and 3. This process also lines up the other end of the complex where the amino acid is linked, up in this region up here, okay? This occurs in what's called, so it first occurs over here in what's called the A site, which stands for amino acyl. That means where it's coming in and binding the correct codon. Within the larger subunit of the ribosome, up on the top, the amino acid gets released from the tRNA and it gets added to the growing amino acid chain, which in this case is called a polypeptide. The ribosome adds the next amino acid to the polypeptide chain by creating peptide bonds that covalently link each amino acid to the previous amino acid in the growing chain. And this occurs in what's called the P site, in here in the middle region. And as you see, what I'm showing is this gets detached and added to the growing chain of amino acids. That's the P site, or that stands for peptidyl. The uncharged tRNA, which has its amino acid removed, is then moved to the E site over here, which is the exit site, where it's released to be recharged with another amino acid of the same type. And then the ribosome moves along to the next codon and decodes it, thus repeating the process of matching the tRNA/amino acid complex with the correct codon. This process just keeps getting repeated over and over and over until it reaches a stop codon. And then the ribosome will disassemble and fall off the mRNA. So the two subunits will come apart at that point. So this process of recruiting the appropriate tRNA/amino acid complex, lining them up, releasing their amino acids, and setting them free again, while peptide bonding each amino acid to the previous one, happens, like I said, similar to a conveyor belt going through an assembly line. The mRNA is continuously fed through the ribosome to keep the process moving along the mRNA to the next codon in line. And as it does, the tRNA/amino acid complexes are transitioned through the different sites on the ribosome, through the A, P, and then E sites. What that does is it ensures that each step doesn't interfere with the next one, and so that each step happens at the appropriate time and at the appropriate place. So once a linear protein is produced, it's called its primary structure. It undergoes some post-translational modifications that affect its function and stability. Most importantly, proteins undergo a folding process where they'll take on their mature, functional 3D structures. The folding of newly synthesized linear proteins into their functional 3D structures takes place via two classes of proteins called chaperones and chaperonins. The chaperones bind the newly synthesized linear proteins and prevent them from folding inappropriately on their own. and they also transport them to the chaperonins. The chaperonins encapsulate the linear proteins, fold them into their correct 3D structures, and then release them. Now, what actually goes on inside a chaperonin capsule and how they perform their job of folding proteins is still a mystery. However, without chaperonins doing their job and correctly folding the newly synthesized proteins, the newly synthesized proteins would not be able to perform their given function. Their folded 3D structure is essential for their function. They'd be useless without it. And a good analogy here is that of a key with the proper pattern of teeth being cut into it. If you don't have a key cut to the exact specifications of teeth for the lock it's designed to fit, they'll never work. And it's the same way with proteins. They must be folded into their 3D structure exactly as required, or they can't do their job either. And proteins have lots of jobs. They do the heavy lifting, so to speak, of cells, tissues, and organs in the body. They're enzymes, receptors, antibodies, cytokines, lymphokines. They're hormones, they're transmitters, they're muscle tissue, they're cell adhesion molecules, they're structural subunits of cells and tissues and organs, and on and on and on. Therefore, as with the DNA and RNA, proteins are essential for life. And again, like I did with the transcription, I just gave you about a semester's worth of molecular biology in about, 10 minutes. And I also left a lot of stuff out. But I want to get to the point here. Among the many jobs of proteins, as you're learning, many of them are involved in these essential processes of molecular biology, like the ones I just talked about. They're part of the 300 or so molecules that I've discussed many times that are required for the life essential processes of molecular biology. Now, I only mentioned a few of them here today, but included in there are the many ribonucleoproteins, along with the ribosomal RNA molecules that assemble to create the ribosomes. For example, the smaller subunit is made up of one 18S ribosomal RNA and 33 proteins. The larger those are all, by the way, 33 unique proteins. The larger subunit is made up of three subunits: a 5S ribosomal RNA, a 28S ribosomal RNA, and 5.8S ribosomal RNA, along with about 50 proteins. There's also the over 20 transfer RNA molecules that the ribosomes utilize. So you should be able to see that all of these molecules are critically important for these processes, and therefore they're critically important for life. With any of these molecules missing, critical parts of these processes won't happen. And if any of the steps in these processes don't happen, life can't happen. Therefore, as I've explained before, all of these molecules have to be there at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed together from day one, which means they simply cannot arise via evolution. And as I reviewed in the previous couple videos, this again is also another example of irreducible complexity to the utmost. So I'm going to end this video here, but come on back for the next one. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching, and as always, we'll leave the light on for you.